So from the AAV capsid side, what we want to do is engineer novel capsids that can penetrate into the brain. It's a challenge, we know this. So the blood-brain barrier keeps out a lot of things from our brain. That's a good thing because it's what keeps us healthy. But finding ways to get around that in order to transduce the brain cells and deliver the ZFPs where they need to go is really important. Luckily, there's a few different ways that this can happen, and I've seen some of this at the conference, which is very exciting too. Sangamo scientists developed a novel platform that I'm really excited about. It's called SIFTER, so Selection in Vivo for Transduction and Expression of RNA. And what's really exciting about this part is the expression. We know that AAV can travel around the body, but it's not enough just for it to get to the target tissue. It also has to get inside the cell and express the therapeutic payload. In our case, it's the zinc fingers. For the SIFTER platform, the scientists developed kind of two parts of that platform. One is using directed evolution to modify the AAV capsid sequence in a way that enables them to generate millions and millions of AAV variants. But what do you do once you have those millions of variants? You need to test them in some way. So using an in vivo selection approach and rounds of this in vivo selection, we're able to narrow down those millions of variants to the top ones that we think could be useful as a therapeutic. And we have a talk today by Dr. David Ojala, who is really excited to share some of these two new novel capsids that he discovered using the SIFTER technique. These ones are administered via the CSF, so a little bit of a different, rather than a peripheral administration, directly into the central nervous system. We think this has a couple of advantages. One of them is that it could possibly evade neutralizing antibodies that are present in the serum. That may be more of a challenge when you do an IV administration. And another thing is that we may be able to use a slightly lower dose. And although everything that goes into the CSF does eventually make its way into the liver, you may be able to limit some of that peripheral exposure by administering in the CSF. Tomorrow we have another poster by Dr. Matt Tiffany on the team who will present some of the first applications of the SIFTER platform to our blood-brain barrier program, where we are looking to deliver intravenously in order to achieve widespread brain delivery. So as I mentioned today, we'll see some exciting data from a non-human primate where we see expression of the AAV capsid is even up to 100-fold better than AAV9, which is what is currently being used in the clinic. We believe this will enable some novel therapeutic applications. We're really excited to be able to leverage these capsids going forward. But we're not done yet. We think that it's not going to be a one-size-fits-all approach. It's likely that we'll need different delivery solutions for different therapeutic indications. We need to keep iterating on the current sets of capsids that we have and continue the program with the blood-brain barrier to cross the blood-brain barrier, because we think that's another way that we may be able to achieve widespread brain distribution. In addition, we would think about other components about making the drug, like manufacturing. You may have a capsid that performs very well in the brain, but you need to be able to make it in order to generate the therapeutic that will ultimately go to patients. That production process, the manufacturing, needs to be efficient and safe, and that's something that we also take very seriously when we're developing these novel capsids. I'm excited to see how we're able to use the zinc finger technology across a wide array of neurological diseases with our partners, Biogen, and also our colleagues at Novartis. With Biogen, we have multiple programs on different diseases, starting with tau for tauopathies and modulating synuclein for Parkinson's disease and other synucleinopathies. With Novartis, we have programs that are aimed at restoring gene expression in diseases of haploinsufficiency, so specifically ones that cause autism spectrum disorder and intellectual disability. It's been really exciting to see how we can activate specifically those genes where the expression is reduced in order to restore their healthy gene expression. We look forward to forthcoming programs with those partners as well, and being able to talk a little bit more about that. In our internal pipeline, we have some great programs going, and we'll be presenting at further conferences this year on those ones.